In our last section of chapter 3, we're going to talk about experimental design. So there's two advantages of an experiment over an observational study. One, we can study the factors we are interested in while controlling the effects of lurking variables, which I'll talk about in more detail later. And two, experiments allow us to study the combined effects of several factors. So when you're designing an experiment, you need to have two things, your factor and your level. So the factor are your explanatory variables, specifically in an experiment. And the level is the various groups the factors take. So for example, factor versus level. So if an experiment compared the drug dosages of 50 milligrams, 100 milligrams, and 150 milligrams, then the factor drug usage would have the three levels, 50, 100, and 150. But the number of factors, the number of levels of a factor, we don't want to get confused. So drug dosage is one factor consisting of three levels. Okay. So there are four critical elements of an experimental design. And number one is comparison. So we want to make sure we're using a design that compares two or more treatments. So we need to make sure that the groups we're comparing don't differ greatly before the experiment begins, or bias can result. So to avoid this, we use randomization. So it is probably the most important element of any experiment. So it has to be incorporated either in the selection process of your experimental units and or the distribution of experimental units into treatment and control groups. So for our purposes, we can use our calculator, the random digit table, names pulled out of a hat, flipping of a coin to randomize an experiment. So a couple things about randomization is we can only compare when the groups are as similar as possible before the treatment is applied. Randomization produces groups of experimental units we expect to be similar in all respects before treatment is applied, and therefore measured differences must be due either to treatment or by chance of random assignment. The next one is control. So in any good experiment, you typically have a control group. So a control group is treated identically in all respects to the group receiving the treatment, except the members of the control group do not receive the treatment. So this control group is the baseline, and the experimental group has only one thing changed, the explanatory variable. So this helps to reduce variability in the response variable. So if one group is controlled, we would expect their responses to be controlled as well. And the last one is replication. Now, replication in an experiment is different than how you may have heard of replication before. So for replication, we have to use enough experimental units in each group so that any difference in the effects of the treatments can be distinguished from chance differences between groups. So in statistics, replication means use enough subjects, whereas in other fields, replication means repeating. So replication here just means we need to have large sample. So even with control, natural variability occurs among experimental units. So we would like to see units within a treatment group responding similarly to one another, but differently from units in another treatment group. So that way we can be sure that the treatment is responsible for the differences. So if we assign many individuals to each treatment group, the effects of chance and individual differences will average out. So there are a few terms that are used in experiments that we're going to see. Some of this you may know. So a placebo is a dummy treatment. So a placebo is usually where they give person like a sugar pill or something, And it's really important, though, that people receiving the placebo don't know they're receiving the placebo. Now, because there is a group that's going to be receiving the placebo, we get something called the placebo effect, which is when subjects receiving the placebo have a response that is similar to what we would expect if they received a treatment. So it basically means that the people in the group think they're receiving the treatment that is going to help whatever you're studying, so they start to do better or have the desired effect that a treatment would have. Next, we have blinding. So this is a very important part in an experiment. It's when the subject does not know what treatment they are receiving to remove the power of suggestion. So like if someone knew they were receiving the drug, their brain might think, oh, it's getting better. So blinding is especially important that people don't know whether or not they're receiving the treatment. And then a double blind goes one step further. So it's an experiment in which the subject and administrator do not know who receives the treatment. So this helps eliminate bias on the administrator's part. So if they know someone personally in an experiment they're performing, they won't give them the drug that they think is going to help them. So it helps to eliminate bias on the administrator's side of things, whereas blinding helps eliminate bias on the subject side. So the design of an experiment is crucial. So experiments are designed with the purpose of isolating the effect of the treatment on the response variable and removing any confounding effects. So in a poorly designed experiment, it might be difficult to tell if the explanatory variable caused a change or if it was another variable that was not measured. So we'll talk about these other variables here in a second. So one of them is something called a confounding variable. And again, we're going to talk about these in more detail. So confounding variables are variables that might affect the outcome, but we did not control or account for in the experiment. And we'll also take a look at some examples as well. So one way that we've already seen the removing of any confounding variables is to randomly assign subjects to the treatment or control group. So this way, any possible bias in the population should be evenly spread among the treatment and control groups. So sometimes, instead of relying on randomization to make groups as even as possible, we actually force the groups to be similar. So there's two things that happen if we have completely randomized design. So the experiment units are assigned to the treatments completely by chance. One of those is that treatment and control groups will be about equal in size. So that's one thing we want to come out of a completely randomized design. The other one is that there are mathematical reasons behind this. Which again, we'll discuss later. So let's take a look at an example. So does regular taking aspirin help to protect people against heart attacks? The Physician's Health Study was a medical experiment that helped answer this question. In fact, the Physician's Health Study looked at the effects of two drugs, aspirin and beta-carotene. Researchers wondered whether beta-carotene would help prevent some forms of cancer. The subjects in this experiment were 22,000 male physicians. There were two explanatory variables, also known as factors, each having two levels, aspirin, yes or no, and beta-carotene, yes or no. Combinations of levels of these factors form the four treatments in the picture. 1/4 of the subjects were assigned at random to each of these treatments. On odd numbered days, the subject took either a tablet that contained aspirin or a dummy pill that looked and tasted like aspirin but had no active ingredient, so that was our placebo. And on even numbered days, they took either a capsule containing beta-carotene or a placebo. So there were several response variables. The study looked for heart attacks, several kinds of cancer, and other medical outcomes. After several years, 239 of the placebo group, but only 139 of the aspirin group have suffered heart attacks. This difference is large enough to give good evidence that taking aspirin does reduce heart attacks. It did not appear, however, that beta-carotene had any effect on preventing cancer. So we want to describe and design a completely randomized experiment here. So for our design, we want to have the random assignment. So what we're going to do is write each of the 22,000 male names on a piece of paper. The first 5,500 will be in the first group, and then you're going to repeat. Okay, so then the next 5,500 will be our second, third, and fourth. So when we are designing our experiment, we always start with our population. So our population in this case is the 22,000 male physicians. And we actually use a tree diagrams. So now are our groups. So we had 4 groups. Okay. And we do want to specify that these were randomly assigned. And then if we know how many specific people were in each group, okay, want to talk about what those groups were. So one of our group took aspirin and beta carotene, and then we had aspirin and a placebo. We also had placebo and beta carotene, and then we had our group that was taking 2 placebos. Okay. Then those 4 groups then led to our conclusion where we compared health outcomes. And I forgot the beta carotene didn't have a placebo. So it's not that they were taking aspirin and a placebo. It was aspirin and no beta carotene. Only the aspirin had a placebo. And then we compared the health outcomes for our 4 groups, and that's how we were able to make the conclusion that aspirin helped reduce heart attacks, but beta carotene had no effect on cancer. And again, because we only looked at 22,000 male physicians, these results can only be applied to males, because there were no women in the study, so we can't talk about the effects or these results when applied to women. So now we're going to talk about lurking and confounding variables. So an extraneous variable is one that is not one of the explanatory variables in the study, but is thought to affect the response variable. So there's 2 types. The first one is confounding. So that refers to a problem that can arise in an experiment when there is another variable that may affect the response and is in some way tied together with the factor under investigation, leaving us unable to tell which of the 2 variables, or perhaps some interaction, caused the observed response. So for example, we plant tomatoes in a garden that's half shaded. We test a fertilizer by putting it on the plants in the sun and applying none to the shaded plants. Months later, the fertilized plants bear more and better tomatoes. We want to know why. Well, maybe it's the fertilizer, but maybe it's the sun. Maybe we need both. So we're unable to conclude that the fertilizer works because any effect of the fertilizer is confounded with any effect of the extra sunshine. So in this case, the fertilizer and the sun could both be variables that lead to more tomatoes. So if we wanted to eliminate the confounding variable here, we would still plant the tomatoes in a garden that's half shaded, but the half that has the sun, half of those would get fertilizer and half wouldn't. And then the tomatoes that are in the shade, half of them would get the fertilizer and half of them wouldn't. That way we can take into account whether or not the sun has any effect on more tomatoes, or if it's just the fertilizer or both. Okay. Then we have lurking variables, which are sometimes referred to as common response. So that's where some other variable drives each of the 2 variables under investigation, making it appear that there's some association between those 2 variables. So for example, there's a strong association between the number of firefighters who respond to a fire and the amount of damage done. One shouldn't conclude that the firefighters may be responsible for the damage. The lurking variable is the size of the fire. So our number of firefighters is tied to the amount of damage, but we also have to determine the size of the fire, because more firefighters respond to larger fires. And if there's a larger fire, there tends to be a larger amount of damage. So lurking variables are more common in observational studies because we see that there's a high correlation between 2 things, but we don't necessarily know if there's something else that is affecting that. In an experiment, the factor under consideration isn't being driven by some lurking variable because we're the ones in control there. So for the examples, We're going to do these in class. So if you want to try and do them on your own, go for it, but we will do the rest of these notes in class together.